Okay, so what have we done so far now? We've done our introduction, we've done our reception, and we went even into detail, especially about the G proteins, but did mention all the other receptors. The intracellular one, how it has to be small and oily, the ion channels with the gate, how it has to open, and the tyrosine kinases, how they're like the multitaskers. So our next thing is to do transduction. Now, if you look at what I have here, I just have two words here, phosphorylation, dephosphorylation, sound like they're opposite, and they are. And then I have secondary messengers. Okay, so what do I have here though? I have a nice little diagram for you. Okay, now I always like to say transduction. Well, a lot of scientists out there call it the cascade of reactions to get eventually to the response. You have a reception that we've already talked about. Now you got to get to the response, but there's a lot of steps in between. How does that happen? That's kind of like whisper down the lane. The reception would be like the person who has the message. He whispers it, whisper, 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 whisper. And then the end person always says what the final thing is. And they never get it right, right? I like to also say it's that whisper down the lane thing or falling dominoes, okay? But a lot of science books will say this cascade of events. Okay, and a cascade is like falling water or something. All right, so what basically happens is you go from one molecule that's inactive and then it's activated. Then it's deactivated. So the next one that's inactive can become active. Then the next one that's inactive becomes active. Then the next one that's inactive becomes active. And each time these active ones have to be deactivated, okay, before you can make the next inactive one active. Now, how does this all happen? There's basically two things, phosphorylation and dephosphorylation. But there are others, okay, and we'll get to the also, those secondary things. Phosphorylation, you know this already. That's when a phosphate attaches to a molecule. Okay, and we know if a phosphate attaches to something, something's going to happen. It gives it like some power to do something. So phosphorylation, I always want you to think active molecule. Okay, but this is caused by these enzymes called protein kinases. Kin means active or motion, right? So think they're going to have it become active. So these enzymes called protein kinases are going to perform phosphorylation on a molecule. In other words, they're going to activate that molecule. They're going to add phosphate. It's kind of like Leo says, GR. This is GR, the gain the electrons, but they're gaining a phosphate this time. So it's a type of reduction in a way, okay, because electrons are going along with it. So here's this inactive protein. When a protein kinase comes along and transfers a phosphate to it, then it becomes active. And remember, it has to transfer one of those little phosphates. And I always draw them like with a P, with a little O. So when something's active, it has a phosphate group attached to it. But then that phosphate group has to be pulled off of it. So it has to be lost, like electrons can be lost, oxidized, right? Has to be pulled away from that. And that's what dephosphorylation is going to be when we inactivate. So if this is molecule 1, right, it starts off inactive, gets phosphorylated by the protein kinases, and becomes active. But then it has to be dephosphorylated by phosphatases, another enzyme that's going to deactivate that molecule 1 there. Then it can take that protein and it can say, oh, you're inactive? We'll take that phosphate and it'll attach that. And that's the second molecule. And then the third molecule has to be activated. And like this little cascade, they'll kind of pass this little phosphate. They attach it, then it has to become detached, then they attach it, then it has to become detached. So the attaching is the phosphorylation. The detaching is the dephosphorylation. Phosphorylation is done by enzymes out here saying, I'm a protein kinase, I'm going to attach this to you. And phosphatases that are saying, I'm going to take that phosphate away from you. Okay, and that's most of the time that's going to cause this cascade of events, changing from molecule 1 to molecule 2 to molecule 3 to molecule 4 until we finally get to the desired effect. But also, there are two other molecules that are worth mentioning, kind of a third one here too. Cyclic AMP, they sometimes represent it as CAMP, and they call it CAMP, but it's a little c, all capital letters. There's also another one called cyclic GMP, just like ATP, GTP, same kind of idea. It's cyclic because the bonds like kind of go back on itself. All right, but it's AM. So it's lost two of its phosphates and it has this bond coming back on itself, but it becomes like a messenger molecule. So sometimes you might not even have this happening here, and instead it's all cyclic AMP, being that second molecule. Okay, and then we call it like a secondary molecule. It can be in the third position or the fourth position as well. Calcium ions also do that. So sometimes instead of having molecule 1, molecule 2, molecule 3, it might be molecule 1, cyclic AMP, molecule 3, molecule 4, calcium ions, molecule 6, molecule 7. Okay, so this cascade can be many, many steps long too. All right, and that's it for the transduction. It's pretty easy there. Okay.